Hello. Hello, I'm Dr. Mirko Pegoraro, and this is your second lesson of developmental biology. This lesson will be actually quite short. It is short because I realized that the day is actually long for you, so the second lesson is a bit shorter than the first one. So what are we talking about today? Today we are going to talk about cleavage, which means cell division. So during cleavage, the cytoplasm of the first cell, the zygote, will be divided in many cells. Then we will talk about gastrulation, which is the movement of these cells to form the free embryonic layer, the external one, the ectoderm, the middle one, the mesoderm, and the internal one, the endoderm. We also are going to talk about the formation of the primitive groove and the node migration and how important the primitive groove and the node are for the migration of cells and the formation of the three embryonic stem layer. So this morning we talk about gametes and fertilization. So we talk about the formation of the gametes and how the gametes come together to form the zygote. Next lesson, so next week, we will have a lesson on the anterior-posterior axis formation, neurulation, not epidermis. We are not going to talk about epidermis. So let's start talking about cleavage and gastrulation. Cleavage is defined as the rapid cell division that divide the cytoplasm of the zygote into many different cells. Gastrulation, on the other hand, is the displacement of these cells around the embryo to form the three main embryonic layer. The ectoderm externally, the mesoderm in the middle, and endoderm internally. During cleavage and gastrulation, we have also the formation of the main body axis. And during cleavage and gastrulation, cell fate is also determined or start to be determined as well. One of the important things about the formation of the free embryonic layer is related to the fact that the free embryonic layer are responsible to produce different things in the embryo. The ectoderm is responsible for the formation of the external surface of the animal, so the epidermis, but is also responsible for the formation of the brain and the neural tubes, so the spine and the nerves. The mesoderm is mostly responsible for the formation of muscles and bones, while the endoderm is mostly responsible for the formation of the internal organs like the gut. When we talk about axis in the embryo, I find it useful to think about mammalian species, vertebrate in general, and humans as they were like a fish. So here you have a picture of a human in the same position of this picture, or kind of the same position of this picture of a fish. So in an embryo, the anterior-posterior axis is defined as the one that goes from the top of the embryo, so the head, to the bottom, to the feet. Or if you want, if we were talking about a fish, the anterior-posterior axis will go from the mouth, so the anterior part, and the fin tail. So this is the anterior-posterior axis, or from the head to the feet. The dorsoventral axis goes from the back to the belly. So that's the dorso. The dorso represents the back, ventral represent the belly. So the dorsoventral axis. And then the easiest one is the bilateral axis that goes from left to right. So this morning we talk about the sperm maturation and the journey of the sperm to reach the egg. We talk about the egg maturation. We talk about sperm capacitation, block of polyspermy. We mention zygote formation and genetic fusion. So this is where we are in terms of embryo formation. So at the moment we have genetic fusion, we have a zygote, and this zygote start to cell divide. So the first thing that's happened where after that we have genetic fusion is cleavage, so cell division. So remember that fertilization happened very high in the ampulla, and then the embryo start to move down the ampulla toward the isthmus and toward the uterus. In this process, the cumulus is extremely important, and also the zona pellucida, because it avoids the embryo during this voyage from high the ampulla down to the uterus. It avoids that the embryo will bind to the surface of the isthmus, for instance, causing an extrauterine pregnancy, which is very dangerous for both the embryo, the developing embryo, and the mother. So during this travel, after fertilization, this movement of the embryo down the ampulla, the embryo start to cell divide. The first cleavage in humans happen more or less 1 day after fertilization. And this is only in humans, not in all vertebrate. The position of the first cleavage, the position of the first cell division, so where it happens, depends on the place of fertilization. So where it depends on where the sperm enter the egg. Now let's have a look on the mammalian cleavage. Mammalian cleavage is quite weird compared to other vertebrate cleavage, and there are a few things that we want to consider. We want to have a look specifically in terms of how different the mammalian cleavage is compared to other vertebrates. First of all, mammalian cleavage tend to be very slow. So the cell division are very slow, and it takes 12 to 24 hours for one cell division to occur. And this is not always the case. In other vertebrates, this is not the case. It tends to be much quicker. The other strange things is the orientation of the blastomers. The blastomers are the cells that are formed during cleavage. So during cell division, the cells that are formed in the embryo are called blastomers. So mammalian cleavage happen. Mammalian cleavage is a rotational cleavage. So it is defined as a rotational cleavage. And why is that? So if you look on this picture here, you will see on the left the first two planes of cell division for an amphibian. So both cell division are perpendicular to each other and are both meridional. So they both divide the cells in a left, the original cell, in a left and right cells. On the other hand, for mammals on the right-hand side, you see that after the first cell division, the first cleavage plane, which divide the cell meridionally in a left and right cell, the second cell division is different for the two daughter cells. One of the two daughter cells divide again meridionally, producing a left and right cell, while one of the other cells divide equatorially, producing an up and down cells. So you see that some of the cells are dividing meridionally, some of the cells are dividing equatorially. And in each cycle of cleavage, which cell divide meridionally and equatorially change. So the cell that in one cycle divide meridionally, they will divide equatorially in the next cycle, and the other way around for the other cells. So this creates a kind of a complex orientation of blastomers to each other, which usually is not seen in other vertebrate, like fish or chicken. Chicken is also complicated, but not in the same way. But birds, amphibian, and reptiles have a much simpler orientation of the blastomers compared to mammalian cleavage. So because of this strange orientation of the blastome, because of this rotational cleavage, the embryos become asynchrony. So the cell division become asynchronous. What does it mean? It means that some cells divide earlier than others, and this results in embryo that time to times have odd number of cells instead of even number of cells. And finally, one other thing that is quite peculiar on mammalian cleavage is that the genome of the embryo becomes transcriptionally active quite early, already after just 3 cell division. So when the embryo is just 8 cells. So when the embryo has only 8 cells after 3 cell division, yes, 3 cell division, the genome of the embryo become already transcriptionally active. And this is peculiar because in other species that's not the case. So, but why is that the embryo of our species and other mammalian species become transcriptionally active so early? Well, it is because by the stage of 8 cells, the embryo undergo a process that is called compaction. So here in this nice picture from A, B, C, and D, you see the different stage of cell division, which are also represented in this sketch below. So you have a zygote, and you can identify their polar body. After one cell division, we have 2 blastomers. A second cell division, we have 4 blastomers. And then on another cell division, we have 8 blastomers. So we have an embryo with 8 cells. So initially, the 8 cells are kind of globular, rounded, but then they start to express a protein that is called E-cadherin. E-cadherin. E-cadherin is like a cellular glue and make the cells to bind to each other very tightly. So at the beginning of the 8 cell stage, the blastomer are loosely attached to each other, but after they start to express E-cadherin, they become very compact and they bind to each other very tightly. Also, they become quite flat. So they form overall what look like a football ball. The next step of cell division produces 16 cells, of course, and the embryo at 16 cells is called a morula because of the shape is reminiscent. The shape is reminiscent of some kind of berry. Now, this is a very important stage because at the stage of morula already we can recognize 2 types of cells. So until now, all the cells were totipotent. They were all equal to each other. They were expressed, meaning that they were expressed in the same genes. But at this stage, already at the stage of morula, we have a differentiation of 2 groups of cells. One internal group of cells. You can, if you want, you can identify on the cells that are internally and not exposed to the surface, or if you want, not exposed to the surface. This internal group of cells is called inner cell mass, or ICM. The inner cell mass are the only cells in the embryo that are going to produce the fetus later on, while the other cells that are not part of the inner cell mass are going to produce tissue that sustain the growth of the fetus later on. So the rest of the cells that are not part of the inner cell mass are called the trophoblast. The trophoblast are the external cells in the 16 morula embryo. So the embryo become a blastocystis at this stage when it starts to produce the blastocoel or the blastocystic cavity. So the trophoblast start to produce liquid that fill a cavity that push the ICM toward one side. So the ICM is pushed toward one side of the embryo, and it is very recognizable in this cartoon here, because the ICM are these yellow cells that are pushed to one side, while the trophoblast is the blue cells that surround the ICM. So the more liquid the trophoblast is producing, the bigger the blastocoel or the blastocystis cavity is becoming, and more toward one side the ICM is pushed. Now, the ICM, I want to remind you again, is the one that produces, you can see here in yellow, the ICM is the one that produces the tissue of the fetus, while the trophoblast is responsible for the formation of the chorion, extra embryonic tissue, and the placenta. Excuse me. So, the trophoblast and the ICM are no longer totipotent cells, because at this point they definitely cannot do the same. They cannot do the same things, and they are expressed in different kind of genes. So let's have a look on what genes the trophoblast and the ICM are expressing to be differentiated from each other. So until the 8-cell stage, all the blastomeres are expressing the same pattern of a transcription factor. And in particular, all cells are expressing OCT4 and CDX2. However, later on, when the blastocyst is produced, the two types of cells, the ICM cells and the trophoblast, are expressing different transcription factor. The ICM is expressing OCT4, SOX2, and NANOG. This OCT4, SOX2, and NANOG form a positive feedback loop. So if these are the genes of OCT4, SOX2, and NANOG that express the protein NANOG, SOX2, and OCT4, this free protein drive their own transcription and also drive the transcription of the other two. So OCT4 is controlling its own transcription, promoting its own transcription, but is also controlling and promoting the expression of SOX2 and NANOG. The same is valid for SOX2, and the same is true for NANOG as well. On top of it, NANOG inhibits the expression of CDX2. So now the blastocyst, in the blastocyst, the ICM cells are expressing OCT4, SOX2, and NANOG, and they are not expressing anymore CDX2. On the other hand, the trophoblast cells do express CDX2, and CDX2 is repressing the expression of NANOG and OCT4. So the two groups of cells are now differentiated not only anatomically, not only in their position, but most importantly in their expression profiles. Okay, so here we have when we have the formation of the blastocystic cavity, we have the process of cavitation. What it means? It means that this cavity, this blastocystis cavity is formed and then filled up with fluid from the trophoblast. When the cavity becomes quite big and the blastocoel is quite large and push the ICM toward one side, at one point we recognize two types of cells in the ICM. So one group of cells of the ICM that is not in contact with the blastocoel or the blastocystis cavity, and this is called the epiblast. The epiblast. So the epiblast are the cells of the ICM that are not in contact with the blastocoel or the blastocystis cavity. Then there is also another group of cells of the ICM that is in contact with the blastocoel or the blastocystis cavity, and this is called the primitive endoderm or hypoblast. The primitive endoderm or hypoblast. So at one point, when the blastocystis cavity becomes big enough and push the ICM toward one side, we can recognize two kinds of cells. One layer of cells that is in touch or is in contact with the blastocoel itself, which is called the primitive endoderm or hypoblast, and one internal group of cells that is not in contact with the blastocoel or blastocystis cavity, which is called the epiblast. Now, it is only the cells of the epiblast that are going to form the future embryo. So far, the embryo have been developing inside the zona pellucida.